Okay, welcome to this webinar from the Kalim Institute about innovative therapies combining several bispecific antibodies. I will be a moderator for this webinar. I'm Michel Cognier. I am myself an immunology professor in the Faculty of Medicine of Rennes University and the University Hospital, working on B cells, B cell genetics, and B cell malignancies. Laurie?
Great. I'm Laurie Sen. I'm a hematologist and oncologist in Vancouver, British Columbia. I'm the chair of the lymphoma tumor group here, so I am interested in all things lymphoma. I have the pleasure today of introducing our illustrious speaker, Dr. Christian Klein. Dr. Klein performed his PhD initially at the Max Planck Institute of Biochemistry, and he focused on the structure of p53 tumor suppressor proteins and network. He has been at Roche since 2002 when he started as a postdoctoral fellow in the Therapeutic Protein Initiative in Penzberg. He, since 2010, has been the head of the oncology program and the department head Cancer Immunotherapy Discovery at Roche Innovation Center in Zurich. He has also been the site head for the Roche Innovation Center in Zurich. He also acts as a lecturer at LMU Munich and the University of Zurich. So during his time at Roche, he's made tremendous contributions and discovery in numerous areas of therapeutics. On a personal level, he has been intimately involved in the discovery of Gazyva and now glofitamab, which I am extremely excited to say was actually approved by Health Canada, first regulatory body in the world to actually approve glofitamab just in the last week. That rarely happens, but we're going to take the win for us. So we're excited to see that really shortly entering the clinic. For his work, he's received numerous awards, which I won't take the time away from his lecture to name, but he'll be presenting today on a very important topic, and really, I think, one of the most exciting areas of clinical development right now across hematologic oncology, but across cancer in general. And he'll be talking about T-cell bispecific antibodies, particularly glofitamab, and the combination with costimulatory bispecific antibodies, including CD19-41BBL and CD19-CD28. So with that, I'd like to invite Dr. Klein to go ahead and begin his lecture.
Yeah, thank you very much, Laurie, for the kind introduction. And we work since a while on CD20 antibodies, and I think it's great to be introduced by you. So with that, of course, I also want to thank here really for the opportunity to present our work on glofitamab and co-stimulatory bispecific antibodies. And let me start maybe with a very short introduction what bispecific antibodies do. They are, as the name says, they are bispecific for the tumor antigen that is expressed on the surface of a tumor cell and to part of the T-cell receptor, here particularly the CD3 epsilon chain that is part of the T-cell receptor complex. And once this bispecific antibody binds its tumor antigen, it has the ability to cross-link the T-cell receptor and initiate downstream signaling, resulting in T-cell engagement and activation and killing of the tumor cells by the cytotoxic granules. It also results in the proliferation of T cells at the site of activation, and of course also the release of cytokines and chemokines that then can recruit additional T cells and immune cells into the tumor. And generally, these T-cell bispecific antibodies, they are very potent with EC50 values in the picomolar to even femtomolar range. They can mediate serial killing of tumor cells, meaning one T-cell can kill several tumor cells at a low effector to target ratio. It's, of course, in contrast to the endogenous immune response. It's independent of a peptide MHC. recognition, and as such, these TCBs, they can also overcome certain escape mechanisms of cancer cells. And and because they recruit polyclonal T cells, it's independent of their specificity and also to a certain extent to their activation and differentiation status. So that's depicted here now. So these potential mode of actions of a T cell bispecific antibody, on the one hand, really the direct killing through recruitment of polyclonal T cells, and they can then also expand these T cells. I think there's still discussion on or investigation going on to which extent can a TCB then also rely on antigen-specific T cells in the tumor microenvironment. And it may ultimately also help to prime a tumor-specific T cell response in the tumor microenvironment by releasing tumor antigens, neoantigens that then can be taken up by antigen-presenting cells. And I think as Laurie already alluded to, this has been a field of very active investigation during the past years. On the one hand, of course, we have all the success with the CD19 T cell bispecific, CD19 CAR T cells. On the other hand, there was a first CD19 T cell bispecific approved, blinatumomab, and now more recently, you also see the T cell engagers, T cell bispecifics really arriving in the clinic with antibodies like mosunetuzumab, glofitamab, but also in multiple myeloma, teclistamab, and even in melanoma, there's also with Kimmtrak also first T cell engager approved. And as you can see here, there is various bispecific antibody formats here in develop, starting with the BITE format, with what is a very short-lived format, then going over to the conventional IgG-based bispecifics like mosunetuzumab or teclistamab, and then the so-called 2+1 T cell bispecific that we are working on. And in the future, there may even be tri-specific antibodies. So we have designed this kind of unique format, so-called 2+1 TCB, because it has two binding moieties for CD20 as a tumor antigen and one for CD3 epsilon, so that it cannot activate the T cell receptor just by binding in a bivalent fashion. That's why we only have one binding site there. And as you can see here in this electron microscopic picture, we chose this format because this additional Fab fragment that we have here attached to this bispecific antibody gives a lot of flexibility to the T cell bispecific antibody, and as such helps to form an optimal killing synapse. It really brings the target cell and the T cell in very close vicinity. On the other hand, by being bivalent, we also have the advantage that we have better tumor targeting and retention. And as you will see later, we can also combine such an antibody with a conventional bivalent antibody, such as obinutuzumab, for example. In order to exclude effects mediated by Fc gamma receptor binding, we have introduced here mutations in the Fc part, so-called PG-LALA mutations, so they cannot bind to Fc gamma receptors any longer. And the most advanced of these 2+1 T cell bispecifics is glofitamab, which is in phase 3 clinical trials, but as you have heard, has recently received the first regulatory approval in Canada for third-line relapse refractory DLBCL. I just wanted to show you here a comparison of what we call a conventional 1+1 IgG bispecific antibody here in blue that you can see depicted here, as compared to a 1+1 where we have the two Fab fragments in a head-to-tail order and the 2+1 format. And you see these formats with the head-to-tail orientation, they are here for this CD19 target much more potent than the IgG format as a consequence of this steric close vicinity that these two Fab fragments can mediate. In the in vitro assay here for CD19, We even don't see an advantage of the 2+1 format. I think that's not entirely true for all antigens. Like for CD20, you can have this, or you will see this advantage. And in addition, of course, also in an in vivo setting, when it is about tumor targeting, accumulation, retention, the bivalent binding provides you with an advantage. So yeah, I think now we are coming to glofitamab, and I think this molecule is designed, as I have pointed out. We have here as a CD20 binder, we are using the variable regions known from obinutuzumab. So that means it's also type 2 CD20 antibody. And we have then here inside of the bispecific antibody the CD3 binder. And as I will show you, this really allows you now to combine this antibody on the one hand with obinutuzumab in order to mitigate the cytokine release. And on the other hand, it may also help you if patient from previous lines of therapy still has residual CD20 antibody levels in the blood. So just a very short reminder, I think you know there's different CD20 antibodies available, such as rituximab, obviously, but then also obinutuzumab and ofatumumab, and most recently ublituximab. And these CD20 antibodies, they recognize this short extracellular domain. They all compete each other because this extracellular domain is very small, but they recognize different epitopes. In particular, we have shown that obinutuzumab binds more to the C-terminal part here of this loop, and that has also very recently been confirmed by cryo-electron microscopy, which really showed the different binding modes on an atomic level that these antibodies mediate. You have here the rituximab Fabs binding to a CD20 dimer, and here you have a single obinutuzumab Fab binding to the CD20 dimer. So and that explains basically also some of the findings that you have, that when you titrate CD20 positive cells with type 1 and type 2 antibodies, you will see double the amount of type 1 antibodies as compared to the type 2 antibodies, because CD20 dimer can basically bind to 2 CD20 antibodies, whereas for type 2 antibody, because of this different epitope recognition, only one antibody can basically bind here to such a dimer. And I think it has really been great to see this now also on a structural level. I also mentioned the P-Chilala mutation. So why are we doing this? Because if such an antibody would bind to the Fc gamma receptor and at the same time to the T cell receptor, this would result in unspecific cytokine release independent of binding to CD20. And for that purpose, we have generated this so-called P329 Chilala mutation that you can see here, that basically abolishes all the Fc-mediated immune effector functions of the Fc portion. And this mutation has now been applied in more than 20 molecules, including approved ones like faricimab and most recently also glofitamab. Yeah, so with that, I also want to show you here some data for the glofitamab as it compares to the 1+1 head-to-tail format and the conventional bispecific IgG-like format. And you can also see here, this is data published here in this publication in Clinical Cancer Research, where you'll find more details, that basically the 2+1 format here in terms of potency really provides here an advantage over the 1+1 head-to-tail and the 1+1 IgG-like format. We could also show that this molecule is then very potent in depleting primary cells obtained from NHL patients. When we test these T-cell bispecific antibodies, because they are specific for the human immune system, we rely on a very specific model system. We are using humanized mice. So these mice, they basically get a bone marrow transplant with human CD34 positive stem cells after they received treatment with busulfan, and then we let these mice sit for several weeks. And during that time, they then develop an immune system that is kind of B and T cell biased. So there's not a big variety of myeloid cells here, as you can see it here in comparison to human blood. See here the different lymphoid tissues. So you see there's a lot of B cells in these mice, as well as T cells. That is really the dominating fractions of the adaptive immune system that expand in these mice. And as such, this model really provides, I think, a very elegant and also translatable model for the testing of T-cell engaging therapies, such as the T-cell bispecifics, but also co-stimulatory molecules. And this is really different from models where you use PBMC in immunodeficient mice, because these PBMC, they will exhibit graft versus host within 2 to 3 weeks, whereas in the humanized mouse setting, you do not observe graft versus host and unspecific T-cell activation. So when we started then initially to test glofitamab in these humanized mice, we were testing them here just in non-tumor bearing humanized mice, and we're looking into the B-cell depletion. And you can see we can observe very deep and potent B-cell depletion in the spleen and the lymph nodes of these mice upon treatment here. And what you can see here is upon the second administration and 3 days after that second administration, you basically have depleted all the B cells in the lymphoid tissues of these mice. This is also related to when you now test this in human DLBCL engrafted or xenograft engrafted humanized mouse model. We see here a very potent dose-dependent anti-tumor response. As you can see here, we were testing here 3 different doses, and starting from the 0.15 mg per kg, where you already can drive the majority of tumors into remission in this very aggressive DLBCL model. We see this remission, and it can be further enhanced if we administer the 0.5 mg per kg dose in these mice. As you may know, these T-cell bispecifics, they have also been seen to induce in patients release of cytokines. And in fact, we can also see here in these mice that obviously for the 0.15 mg per kg, we really start here then also to get significant release of cytokines or secretion of cytokines, the typical ones, TNF alpha, IL-2, IL-6, interferon gamma, and the other ones stated here, that you observe here 24 hours after the first injection of the bispecific antibody. So at that time, we were reasoning, how can we maybe reduce the cytokine release, and we were thinking here that we could combine it with obinutuzumab, because this antibody has 2 binding sites for CD20, and as such, it has the same binding affinity like an antibody, and you can imagine to combine these 2. And if you do this with a conventional 1+1 bispecific antibody, it's difficult for this antibody to compete with the bivalent antibodies, because they bind with avidity to CD20 and basically block the access. But if you have a 2+1 antibody that has the same affinity, it can basically compete with obinutuzumab and then bind to the B cell and subsequently recruit T cell for killing. And yeah, just to keep in mind that in order to see potent killing with such a T cell bispecific antibody, you only need very low occupancy of CD20. So let's say in the range of 1% occupancy, you will already see potent killing with glofitamab in this setting. So you do not need to outcompete all the CD20 antibodies, but can do this with part of it. So we have then investigated the combination, or let's say pre-treatment with obinutuzumab or Gazyva, so-called GPT. So these mice, they're administered with obinutuzumab, and then we come in a second dose with CD20 TCB or glofitamab. You can see the scheme here. So of course, in terms of anti-tumor efficacy, we do not see here any negative impact, as you can see it. This is a TCB alone. This is Gazyva, which is not able to control this WSU-DLCL2 tumors that are very aggressive, as you can see it here, very rapid progression. But now if you initiate the treatment with Gazyva and then subsequently treat with a CD20 TCB, you also see here the very potent anti-tumor efficacy. The important aspect now is, of course, what happens to the cytokines. And you see here, if you inject glofitamab into the mice upon the first injection, you get again the cytokines. They will then be gone upon the second injection, because you will have depleted peripheral cells and also part of the tumor. And then when you give the second dose, you don't see cytokine release any longer. With Gazyva, in contrast, you don't see cytokine release. And if you now give Gazyva first and then come in with the CD20 T cell bispecific, as you can see it here, you see very minimal cytokine release. And that was really the rationale for the development of this strategy, where you first treat with Gazyva and then initiate the treatment with glofitamab. I'm not showing you here the data, but this was also confirmed in cynomolgus monkeys as part of the toxicology package, as it's required for the health authorities. And we could also show there very clearly that with Gazyva we could prevent the cytokine release, whereas already with low doses of glofitamab, without Gazyva pretreatment, you get a significant cytokine secretion. And finally, I think what in these models, what is quite interesting, that if you do not only do Gazyva pretreatment, but you do the combination of these antibodies, and we are combining it here with a suboptimal dose of glofitamab, because otherwise you get the tumor remission. You can see that you can basically, by combining Gazyva with glofitamab at a suboptimal dose, you can basically induce the complete tumor remission here in these two models. So I think that is very supportive data that there is not a negative impact for the Gazyva pretreatment. So you could even in the future think about combinations. And based on these data, glofitamab was tested in a phase 1 clinical study in relapsed refractory B-cell lymphoma, and the data were published in JCO. I think many of you will probably have seen these data, I think, which really show the impressive activity of glofitamab in these relapsed refractory patients. And importantly, when we then also did some biomarker analysis in patients and in biopsies and compared it to what we have seen in the mouse models, the T cell infiltration, T cell activation, I think we see a very similar picture with infiltration of T cells. They will proliferate, they will expand in the tumor microenvironment and form really densely T cell infiltrated areas. At the same time, the tumor will be will go into apoptosis and be depleted. And I also wanted to show you here a few patient cases really from the very early experience with glofitamab in patients. And I think this was as part of a compassionate use program, a 5-year-old boy with Burkitt's lymphoma, CD20 positive from France, that had received 8 different therapies, including CAR T-cell therapy. But the best outcome was partial response and really showed this impressive response to glofitamab. Similar also here, a patient with a heavily pretreated relapsed refractory DLBCL here that you can see here, and he had received also 6 prior lines with chemoimmunotherapy, radiotherapy, and he also achieved a complete response after 6 doses of glofitamab. So I think that was really the basis then also to initiate the pivotal study in relapsed refractory DLBCL patients, and these data have been published very recently in the New England Journal of Medicine, and so it's a pivotal phase 2 study that also are the basis now of the regulatory filings in Canada with the FDA and in Europe, and showing a very high response rate in glofitamab treated patients, as well also durable responses in these patients. And as a consequence, now there's of course a broad development program going on for glofitamab in combinations like with GEM-OX, also in first-line DLBCL, together with R-CHOP and Polivy, and additional studies in NHL. So that brings me now to the second part of my presentation, and because we have seen from CAR T-cell therapies that co-stimulation in T-cells is a very important aspect, and we have developed several agents to be able to combine T-cell bispecific with co-stimulatory molecules, and today I will talk about CD19 4-1BB agonists and CD19 CD28 agonists. So these co-stimulatory molecules, they are known from T cells. So when these T cells get activated, on the one hand, they can express these inhibitory receptors in order to control the T cell response. And on the other side, they also express the activating receptors, including CD28. Well, CD28 is already there, also at baseline, but they rely on it for co-stimulation. Or then members of the TNF receptor superfamily like 4-1BB or CD137 that you can see here. And this is basically then, these T cells, if they get then the co-stimulation from the natural ligands for these co-stimulatory receptors, they basically receive signals that increase their activity, durability, and persistence. And that's, of course, what we also want to achieve. So on the one hand, we have CD28. This is expressed on naive T cells constitutively. It's a more kind of an early co-stimulator that also results in a very strong co-stimulatory effect, as it was as known from the CAR T-cell therapies. And on the other hand, you have the 4-1BB that is typically expressed at low levels on T cells, but then upregulated upon activation. So it's more kind of a late co-stimulator, also preferentially found on CD8 T cells, but also on antigen presenting cells and K cells. So it's also working together with innate immune responses, whereas the CD28 is can be considered as a T cell specific co-stimulator, but that works on CD8 and CD4 T cells. So what's the now? How do we want to, or how do we achieve this? This concept is known from T cell biology, T cell immunology. So T cells receive a signal 1 through the T cell receptor that typically interacts with the peptide MHC complex, but it can also be engaged via T cell bispecific antibody. So when this T cell bispecific antibody binds to the tumor antigen, it will cross-link the CD3, result in signal 1, and then the signal 2, the co-stimulation, in a very similar fashion, can also be provided with a bispecific antibody that binds to a tumor antigen and to the co-stimulatory receptor, as said, CD28 or 4-1BB, resulting then in improved effector functions, improved tumor cell killing, and a better duration of the response. As I alluded to, this was really essential for the success of CAR T cells. The first generation of CAR T cells, it lacked these co-stimulatory domains, and it was not very successful. But then when in the second generation, the CD28 and 4-1BB co-stimulatory domains were included, or they can even be combined. And that was then really essentially the key factor then resulting in the improved efficacy, durability, and persistence of CAR T cells in patients with B-cell malignancies. So I'm showing it here again for the 4-1BB receptor system. That's just what happens in nature. Peptide MHC, TCR interaction, signal 1, and then as a signal 2, you have the interaction of 4-1BB with a 4-1BBL, the ligand that can be found on antigen-presenting cells. And then as a consequence, you have this improved proliferation, cytotoxicity, cytokine secretion, Th1 polarization, contributes to memory formation and the survival of the T cells by being anti-apoptotic, and also serves or protects from exhaustion to a certain extent and improves the metabolic fitness. Of course, 4-1BB antibodies have been clinically developed, and also utomilumab has been tested in combination with rituximab. But generally, the first generation of 4-1BB antibodies that relies on hypercross-linking via the Fc gamma receptors, it was either relatively toxic, such as urelumab, or and then resulting in dose-limiting toxicities, or it was not very efficacious, such as utomilumab, which is a very weak 4-1BB agonists. The field in the past 5 to 10 years has attracted a lot of interest. We have just reviewed this very recently. So there is various bispecific antibodies in preclinical and clinical development to activate 4-1BB receptor signaling. What we have chosen is a slightly different way. We are really using the natural ligand, the trimeric 4-1BB ligand, in a split fashion, attached to an antibody that targets it to the tumor antigen. And you can see this here. So one arm directed to CD19 as a tumor antigen, and then we have here the trimerized split 4-1BBL ligand. And the idea behind this is really now reduce the toxicities by eliminating Fc gamma receptor binding, because that was part of the reason why the conventional 4-1BB antibodies showed hepatotoxicity. Then deliver this into the tumor, and by binding to the tumor antigen, we introduce the hypercross-linking of the 4-1BB receptors. So they will all be cross-linked. They can then phosphorylate, activate the downstream signaling. And this is really truly targeting dependent. That means if this antibody is not bound to CD19, it basically has no effect, even if the 4-1BB is found on T cells. And then only when it's in the tumor, when it binds to the tumor antigen, then it can cross-link 4-1BB and result in the downstream signaling, cross ubiquitination, and different phosphorylation events downstream of this. So this is again here electron microscopic picture. We have here also flexibility again, so which is important, I think, to form the synapse. And yeah, I think here we have chosen another target, but I think it's nice to illustrate here that this bispecific fusion protein then is really able to bind simultaneously to tumor antigen, in this case, well, first of all, 4-1BB, and then it can also bind to FAP, so in a simultaneous fashion. And the same holds true also for the CD19 antigen. So there are also different ways how you can do this. I showed you the FAP molecule, which provides the co-stimulation in trans because it binds to different cells, FAP positive cells. In the case of CD19, we are really talking about cis because you have CD20 binding to CD20 on the tumor cell, and the CD19 is also expressed on the tumor cell. So you really can attack the tumor and now with these two bispecific antibodies, and then we engage the two pathways on the T cell. And when we tested this then in some of these models that I've shown you before in combination with glofitamab, we see very impressive combination effect. So we had also here in the WSU DLC tool model, we used it at a somewhat suboptimal dose. Here, and you see then here the CD20 TCB is not very efficacious. Also, if you combine it with untargeted control of the 4-1BBL fusion protein, you see basically no improved efficacy. But now if you combine it with CD19 4-1BBL, you can induce tumor remission, whereas the molecule per se on its own is not efficacious. Same here in the NALM6 model. You can see this model is really very low in CD20 expression. But again, here when we combine CD20 TCB with CD19 4-1BBL, we can induce remission of these tumors, whereas you cannot achieve this if you combine it with an untargeted 4-1BBL fusion protein. And then when you look at earlier time points into some scout animals, you see we have here a huge expansion of CD8 T cells over Tregs with a combination. And when you look by IHC, here a very massive infiltration of T cells into the tumor that are granzyme B positive, so they are cytotoxic and they start to kill off the tumor here, the CD20 that is here indicated by CD20, CD19 positivity. And when you just do this with a T cell bispecific alone, of course, you also get T cell infiltration, but to a much lower extent as compared to the co-stimulator combination. Also, when you initiate the treatment with CD20 TCB here in this OGLY18 DLBCL model, which is even more aggressive, you will see that these tumors ultimately, after an initial response, they will relapse. They will go into progression again. And when you then initiate the treatment with a CD19 4-1BBL fusion protein, you can drive quite a significant percentage of these animals into complete tumor remissions. We have also applied a very similar approach now to target CD28 with a bispecific antibody, and you can see here the design of this antibody. So we have here high affinity tumor targeting arm recognizing CD19. We have a silenced FC again, and then we have a monovalent CD28 binder that has reduced affinity so that it does not bind everywhere in the periphery to CD28. I think you probably or at least some of you remember the case of TGN1412, the Ticinero molecule, which was a bivalent CD28 superagonist that was applied in healthy volunteers in the UK and resulted in very severe side effects. So there was really a shock to the clinical development community back in 2005, I believe, or 2006. And yeah, we have now found out that the reason, or we and others have found out, the reason is really, on the one hand, the bivalency of the antibody, which makes it a superagonist, and it's binding with high affinity, and at the same time you have the Fc gamma receptor binding of this antibody, in particular binding to Fc gamma R2b, and this can then result upon Fc gamma receptor binding to T cell activation, so-called superagonism. And that's one of the reasons why this antibody exhibited these severe side effects. It was not really in the animal models that the researchers had performed. They did not see this because they basically used inappropriate models. But interestingly, if we test this in the humanized mice, we see that in fact the TGN molecule here with two different backbones, IgG4 and IgG1, results in cytokine release, as you can see it here in red. So that's not the case if you convert this molecule into a bispecific antibody that is monovalent and has no Fc gamma receptor binding any longer. So in this case, even if it can bind to CD19, you basically do not see cytokines. So that's a different time points here, starting from 4 hours to 24 hours and 72 hours. So then we also, of course, were interested in testing the combination of glofitamab with CD19, CD28, and you have here these animals here where you see all the in a disseminated setting where you see the B-cell tumors in these animals. So this is different from the models that I've shown you before, where we had the subcutaneous tumors. Here, this is a model that is more reflective probably of the natural situation with disseminated tumors throughout the body. And you can see that, while CD20 TCB here, and we are using a luminescent tumor cell line, while CD20 TCB on its own has some effect, the CD19, CD28 as a monotherapy does not. But when you combine the two, this is here the red dots, you can also here basically eradicate these tumors at a later time point, which I think is very impressive efficacy. Whereas with the CD20 TCB, while you also have anti-tumor efficacy, you cannot completely eradicate the tumors in these mice. We also see here when we combine glofitamab with CD19, CD28, we see very strong accumulation again of CD8 positive T cells in the tumor microenvironment. So also confirming what we have seen for the CD19 4-1BB agonist. And then similar also to what we have tried with 4-1BB, we have done here also first line, second line setting where the animals received GPT, Gazyva pretreatment, and CD20 TCB, and then when they went into progression, they were treated here with in combination with CD19, CD28, and we can then basically also prolong the response and delay the tumor escape. And finally, I think what we done, of course, one question was now, okay, we have CD19 4-1BB, we have CD19, CD28. How do these molecules differentiate? They look pretty similar when you combine them, or if you combine them with glofitamab on their own. And that's why we now have also tested the triple combination of these three agents. And I think it's particularly interesting that the best outcome we observe when we combine glofitamab first with the CD19, CD28 molecule, followed then by the treatment with the CD19-41BBL protein. And that you can see here where we then are really able to drive the majority or basically all of the animals into complete tumor remissions. That's a little bit reminiscent maybe of what you heard about the CD19 CAR T cells, where there's also the belief that maybe the CD19, CD28 based CARs, they kind of hit stronger in the beginning. They are maybe less prone for the durable T cell persistence. That's where the CD19-41BBL or CD41BBL based CARs may have some advantages. And here, of course, by combining it with off-the-shelf reagents, we really have the possibility to also investigate a triple combination, and that can result then in superior tumor control. And with that, I really want to thank all the contributors. You can imagine this was really many people that contributed here within Roche, but I'm focusing it here on the people that were involved in the discovery of these molecules. So Marina Bakac, she's a research project leader for glofitamab, Sylvia Herter for CD19-41BBL, and Johannes Sam for CD19-CD28. Claudia Ferrara, she really contributed to the 41BBL platform from a technical point of view, as well as Thomas Hofer for the CD28 platform. And then, of course, Pablo Umana, who is heading the research at our site and who started all of this with discovery of GA101 or obinutuzumab Gazyva. And with that, thank you for listening. I'm looking forward to your questions.
Okay. Thank you very much, Christian, for this fantastic presentation. It's really impressive and very promising. So it's time for question. To begin with a question about the format of your antibodies. Could you explain the reason for your choice to have several bispecific rather than combining all these reactivities into single molecules that could associate like tri-specific or tetra-specific?
Like tri-specific, yeah.
That's...
Well, there's for example, I think Sanofi, they have described a tri-specific molecule. I think the challenge with these molecules is, of course, when you have a tri-specific molecule, you have to make sure that they cannot activate the T cell without binding to CD20. Right? And for example, if you have CD3 and a CD28 binder in one molecule, it can already activate T cells without binding to CD20. And I think that's also, I don't know, we have not tested some of these tri-specifics that have been described, but also when I was asking at a conference, you get some low-level T cell activation with such approaches because, you can just bind to CD3 and CD28, and that will then result in some cross-linking of the T cell receptor, and then you get some low-level cytokine release, some low-level T cell activation. Therefore, we believe that it's better to kind of separate these mechanisms because that gives you then really the possibility to trigger one receptor
change
at a time.
the schedule also. That's nice to have one antibody at one time and then...
Exactly. Also, right. Also think about the doses. There may be the dose response of a T cell bispecific, which is a super potent molecule, may be quite different from the dose response of a co-stimulatory molecule. They may overlap, but the optimum may not be at the same concentration. It's also known, particularly for the co-stimulatory molecules, you can observe something like a bell-shaped phenomenon, meaning that if your concentration is too high, your molecule will be basically titrated out of the synapse and the activity will go down again. So that's also something to really keep in mind. And yeah, I think there may be in the future, there may be approaches how you can do this with dry specifics, but you also have to keep in mind that the molecules are presented you today, they were invented several years ago. I think where also the field was just pioneering the T cell bispecific antibody space, right? At this stage, it's only we are talking about 4, 5 approved T cell bispecific antibodies.
But it's very interesting to be able to precisely adapt the dose and the targets at different times. So I agree. But what about the adverse events with combination? For example, if you have non-tumoral mice, humanized mice, are you monitoring some T cell activation, some late T cell activation or autoimmunity in late treatments?
Yeah, so we are, of course, we are looking into this. I think our experience here is that particularly now for the cytokine release, the essential step is really these first infusions. And in the case of glofitamab, they are managed with Gazyva pretreatment and then the step-up dosing. But then once you have reached the step up and the dose, the 30 milligram in that case, and also what similarly in the animal models, once you have reached the efficacious dose level, then the subsequent infusions, they are not critical any longer in terms of cytokine release. I think they will also make sense because, you will already, these bispecifics, they have a very fast onset. And at that point of time, you will already have depleted all the easily accessible B cells that are still in the bloodstream or that are easily accessible from the bloodstream. And then when your cytokines maybe are rather secreted more within the tumor, then you will not have these systemic side effects. Then when it comes to autoimmunity, I think to my knowledge, this is rather something that applies maybe to reactivating the endogenous immune system with checkpoint inhibitors like CTLA4 particularly, and also to a certain extent PD-1, PD-L1 inhibition. But in the case of the T cell engagers, this, to my knowledge, has not been described to be such a major finding in clinical trials. But I think the clinical trials will show, of course, once bigger patient numbers are treated.
And a last question from me before going to the chat. Do you have any idea of the repertoire of the T cells that are accumulating with time after your different combinations? Which T cells are amplified when you are using one bispecific or the other?
Yeah, I think for the T cell bispecific, it's the memory effector subset. I think that is engaged there very, very prominently. In principle, T cell bispecific can engage every T cell, right? even naive T cells or even Tregs. Yeah, but typically it's more kind of the memory effector type that is. That's engaged with T cell engaging bispecific antibodies.
Okay. Maybe Laurie, you have a question, or we can go to.
Just a follow-up question on what you were commenting on in terms of the augmented toxicity with the bispecific. So were you suggesting that these are going to need to be sequenced in, for example, and that if you sort of first treat a patient with the CD3, CD20 bispecific and induce the CRS and get beyond the first cycle, then you would introduce this? Is that what you're suggesting? Or
you mean the costimulatory bispecific antibodies now? I think that is, that was the initial thought, right? That you could do it like this. I think there's currently some discussions on, based also on the clinical profile and how the experience is. I think then the clinical team is really deciding what's the best time point. I think there has not been much communicated. Therefore, there's no published data yet. But I think in general, I think what I can say is that it's not that now the co-stimulation now would really boost the cytokine release, and that may even allow maybe to give this at earlier stages already. Yes.
Okay, so we have a question from Camille Laurent, is asking, have you ever confirmed the presence of TCB in the tissues of your mouse models? Does TCB interact with all T cells, including gamma delta T cells? Does TCB show the same activity with T cells regardless of their maturation stage?
Yeah, so these T cell bispecifics, they can also engage gamma delta T cells. Yeah, that's the case. And as I mentioned, it's primarily the effector memory subset that is engaged here. It's still difficult to say, from an efficacy point of view, which T cells are really now the ones that contribute ultimately in patients to the efficacy. I think the good thing about T cell engaging bispecific is that they can always, they can also recruit new T cells as they come out of the immune system, out of the bone marrow. They can engage them and bring them into the tumor and engage them then locally. So there's you will always have this kind of influx of T cells into the tumor microenvironment.
There was an earlier question as well, which is sort of along the same lines, and it was, can you share your thoughts on balancing T cell activation and exhaustion and finding the right balance for maintaining T cell functionality and persistence?
Yeah, I think that that's an important point. I think colleague of Marina Bakac, she has looked closer into, let's say, the topic of resistance mechanisms and exhaustion, and she will also present a poster at the upcoming ACR meeting. In her hands, the exhaustion does not seem to be a major contributor here in these models that I've shown you. Also the models where we have the relapse. So it may be other factors that contribute. That's still under investigation. But it's not that you now can say, okay, these T cells are exhausted. That's why the tumor is coming back. And also, I think when we compare the combination with co-stimulation and the combination with checkpoint inhibition, which may be rather suited to deal with an exhausted phenotype, I think overall it appears that the co-stimulation is more promising than the checkpoint inhibition. So I think it's really a multifactorial thing, that's probably contributing to the resistance mechanisms. But certainly, I think you have maybe seen there are certain publications showing that in vitro models, when you expose T cells repeatedly to T cell bispecifics and do this kind of repeated killing, then you can get there an exhaustion phenotype induced. Yeah, that's all. So what's the relevance of this for the clinic still needs to be further investigated. I think because probably T cell bispecific does not entirely rely on the tumor resident T cells, but also brings in the new ones. I think that probably gives a certain protection from that.
There was another question from Suman Mitra asking, have you investigated the occurrence of epitope spreading following a BiTE treatment?
Yes, that's a very interesting and very hot question. I think we are still looking into this. I would say in the B cell malignancies that are typically also not Also, there is somehow quite some neoantigens present there, but they are not known to be, let's say, tumors that are typically responsive to endogenous approaches reactivating the T cell immune response. So I think in this case, to my knowledge, there is very little evidence that you would really have epitopes spreading after blinatumomab or after some of the other T cell bispecific antibodies. So I think in order to achieve this, you will probably need additional combinations. For example, boost the antigen presentation, dendritic cell uptake, and so on, the myeloid axis, in order to boost the generation of a secondary immune response and the epitope spreading. But I think for the efficacy, as you see it, probably with these T cell bispecifics, they are to a big extent driven by a direct engagement of T cells. But I think, it's still possible that the epitope spreading there plays a role in the underlying response.
I did have a question. So just thinking of things as a clinician, we've got an expanding number of options that target CD19. You talked about CAR T cell therapy. We have tafasitamab and the concept of CD19 loss and how we're going to sequence these options. I personally don't know enough about CD19 loss and what would be expected to initiate it, or is it outgrowth of cells that don't have CD19? Can you speculate on whether or not that's going to be a concern with these engagers?
I think generally my impression, I don't know how scientifically sound this is, but that generally it appears that the CD19 loss is kind of more prevalent than CD20 loss. And I think from just seeing what's happening with CAR T cells, it may be that B cells, they have a disadvantage if they lose CD20. Of course, there has also been described a certain percentage of patients showing CD20 loss. So that's comparing CD19 and CD20. Now coming to your question, I think we will have to see the advantage that I would see here is that by combining two different antigens like CD20 and CD19, here that may in a way counteract the loss, because you are tagging basically these B cells from two angles with two tumor antigens, and that may reduce the frequency of the loss.
We have a question from Bertrand Nadel about long-term efficacy, and he's asking, noting that similarly to CAR T cells, you have durable response, and what in your mind is the basis for this long-term efficiency with antibodies?
Well, I think maybe like for other approaches in the B cell malignancies, is probably really also the depth of the response, right? Also with obviously with R-CHOP in a very significant proportion of patients, you can also eradicate the tumor, and that's probably related to the really receiving deep CRs in those patients, and not requiring then permanent maintenance treatment necessarily. So I think that's one factor. It could also still be that I think what we were talking about before, that T cell surveillance may also still play a role, that you basically have a certain underlying immune response that contributes then to the long-term control. Of course, different if you talk about bispecific antibodies, if you stop treating the patient, after a few weeks, the bispecific antibody will be gone. That's different from CAR T cells, where these cells can, they still can expand and be present in the patients, and there has been shown that they are present also because they permanently get the CD9 from the bone marrow. They will always see normal CD19 cells that basically can retain them then in the patient. So in that sense, I think we will have to see how compares the durability of the response. And of course, there's also still the possibility to that you would introduce something like a maintenance treatment with a bispecific antibody. I think that's not the case now. Glofitamab has a defined number of schedules that the patients are treated with, and then they are off. But of course, there's also still the possibility then to treat them later on. And if you think about it, some of the challenges that you face with bispecifics, and when you have patients with a very high tumor load and the cytokine secretion could really be managed if you rather go in a minimal residual disease setting, right? If you give such a bispecific in an MRD setting just to eradicate the remaining malignant cells, it may be very attractive, and it could then really help also to give you these very long-lasting responses.
So it's an interesting thought, actually. I mean, you did comment that by themselves, these bispecifics, co-stimulators don't have efficacy when you've got full tumor. I mean, was that a surprise that you wouldn't see any efficacy used by themselves?
So in which setting did you?
Well, I seem to recall that on one of your slides you said that basically the CD19-41BB didn't have any clinical efficacy by itself.
Yeah.
Are you suggesting that in the MRD setting it might have value by itself?
I don't think on its own. I think you would still need to combine it, because the co-stimulatory bispecifics, they really rely on the presence of a signal 1. Of course, if you would have a signal 1 coming from a T cell, that recognizes a neoantigen, a peptide MHC TCR interaction, then the co-stimulation would also boost that signal 1. But as the evidence for this, I think is still limited, I would at this stage still assume that you would need to combine it with a T cell bispecific antibody in order to provide the signal 1.
Just another question, Christian. In fact, with your bispecific antibodies, you virtually, except for the pre-treatment with obinutuzumab, you lose any effect from complement or this classical aspect of the immune response. Don't you think this could be also something which would be worth being preserved in a combination?
Yeah, I think that's
also having a classical effect.
I think that's a valid aspect. I think that's also part of why we have investigated the combination of glofitamab with obinutuzumab, because of course you can with this approach you can engage NK cells, myeloid cells, antigen presenting cells, right? They could also take up tumor antigens, present them and raise a T cell immune response. So I think particular if you think about that aspect, it may continue to be attractive to evaluate these combinations. I think I also have to say, I think there was a phase 1b study combining glofitamab and obinutuzumab, and I think there was also promising activity, but I think not to the extent that you see it in the preclinical models.
Okay.
There's another question from the audience, and the question is, does the 4-1BB bite induce tonic signaling in T cells?
No, it does not, because I think these cells, first of all, they need the signal 1 in order to upregulate the 4-1BB, and then only in those cells that have the 4-1BB upregulated can you activate 4-1BB signaling. As mentioned, if you would have a T cell that via peptide MHC interaction is specific, and then you would give a CD19 4-1BB-L molecule, then you can also, of course, trigger the 4-1BB signaling in those cells. But otherwise, if you don't have a signal 1 and the antibody and you don't have a tumor antigen, or the antibody not bound to the tumor antigen, then you don't see 4-1BB signaling. It's a low affinity interaction, the 4-1BB-L with the 4-1BB, and without the binding to the tumor antigen, you will not trigger the 4-1BB signaling.
From your own manuscript, Christian, I understood that it might be better to have an anti-4-1BB in order to obtain hyperclustering of 4-1BB rather than 4-1BB ligand?
No, in our experience, we compared bispecific antibodies and the 4-1BB-L ligand, and in our hands, the 4-1BB-L ligand gave us the strongest 4-1BB signaling as compared to a couple of bispecifics that we had tested. And then particularly when we tested it in the in vivo setting, this had quite a big impact on kind of the synergy that we could observe for the combination. Well, I would not exclude that there may be bispecific antibodies out there that are also very potent 4-1BB co-stimulators, depending on the epitope, the antibody design, and so on. But just with the antibodies that we had at the time and compared to the ligand, the ligand was the most efficacious one.
So another question from Bertrand. In an ideal first-line DLBCL setting, what in your mind would be the best combo with bispecifics?
I'm going to take notes on this one.
Well, our hope is that by combining T-cell bispecific with co-stimulatory agents, you may in the future be able to not rely on chemotherapy any longer. So I think that would probably be a long-term goal, that to replace chemotherapy with a combination of T-cell bispecifics with co-stimulation. But of course, that still needs to be shown in the clinic in direct comparison that this is a feasible way. And on the other hand, also, bispecific, they can also be combined with standard of care regimens. Now be it CHOP or agents like lenalidomide that also have kind of working via the co-stimulatory axis. So I think all these combinations are very promising in my eyes, and I think we'll see more of those. But hopefully at some point, we can also then, because in the B-cell malignancies, the therapies, they continue to add up if you always add on standard of care, and hopefully at some point we also reach the situation where we are maybe taking off certain of these agents from the combination regimens. It could also in the future you may also think about combining T-cell bispecific antibodies, right? I think, Laurie, you referred to the antigen loss, And of course, you could also think about combining a CD20 T cell bispecific with a CD19 bispecific, for example, or other targets, and that has been is already being done for the CAR T cell therapies, CD19, CD20, or CD19, CD22, in order to prevent antigen negative relapse. And I think that could also be the future in bispecifics.
Just a somewhat related question. I mean, we're going to have several CD20, CD3 bispecifics on the market likely, and people will probably choose their favorite. But I'm assuming that if a patient doesn't respond to one CD20, CD3, it's unlikely that rolling over to another one, even though they have slightly different epitopes, would make a difference, correct? I mean, do you have any thoughts on that?
I think it probably depends on the potency, right? I think there are some differences in the potency and maybe also in the baseline response rates of these bispecifics. So I think one could still imagine, I think, a situation where maybe one T cell bispecific could be more efficacious than another one. In my view, I think that's glofitamab was really kind of designed to be a very potent T cell bispecific by being bivalent, by having this head to tail orientation that allows maybe a tighter synapse formation. So in that sense, it could still provide an advantage, but that would need to be shown. I think it may still be worth the effort, to test this in the future. As particular also if you think about having a CD19 bispecific maybe, and then a treatment with a CD20 T cell bispecific, I think that would make a lot of sense, even more than if you would have a CD20 combination.
Well, probably all the questions have now been done from the audience, so we may conclude. I think that was really a very fascinating and inspiring talk. Thanks a lot. Congratulations for this fantastic work and for all these new antibodies that will be used in the near future. Laurie, if you want to conclude from the clinician point of view.
Yes. Again, I think I cannot hear about this progress enough. So I think it was a fascinating talk for me, and I again can't stress how much this. I always say this, that our biological insight and sort of the progress in biology is translating into clinical progress. But I think this is a clear example of how the field is really benefiting from all of this work that you and other similar scientists in the field have done. But it's just exciting to see that the science is actually proving true in the clinical setting as well. So and how to sort of continue to build on this progress and make them better. I think I'm pretty excited about what you've presented on these costimulatory bispecifics, and really commend you on all of your work. And thank you for an excellent talk.
Thank you very much. I believe that we will really see more of these co-stimulators in the future in the clinic. Be it our ones, be it from other companies. I think that will definitely be one of the big things in my eyes. And I also want to thank you for all your engagement, for all people listening really here on a Thursday before Easter, joining this late evening presentation.
Yes, great. Well, with that, I think we'll close out the session, and hopefully everybody celebrating all the holidays going on right now will have a lovely weekend.
Okay.
Thank you very much.